I'm Bruce Whitelaw. I'm a molecular geneticist and have been since I left university, basically. Fascinated with genes, fascinated about what they are, and have lived the last 30 odd years trying to engineer them in one way or another. And the tools have just got better and better over that time. For one example of gene drive that's been proposed, and we've proposed it as well, has got the exciting name of X-Shredder, all right, you know. And what happens here is that the gene drive, when it's functional, will destroy the X chromosome, and therefore you don't have an X chromosome. If that's in your sperm, then you're only going to transmit Y-bearing sperm, and therefore you're only ever going to produce boys, males. That's quite simple. But if you destroyed the X chromosome in any other cell, it wouldn't function and you would not have an animal. So there are a lot of nuances around the timing of it, exactly which genes you're going to work with, and the functionality of those genes, which you need to have a good understanding of before you can embark on gene drive. But the tools are there to do it. And when it comes to understanding genes, every day there's a new line of mouse that's been produced that's got a mutation in a gene or a knockout of a gene, and we understand more and more. And there's a list of genes which we can work with. And at the end of the day, it's still trial and error. You're going to come up with your candidate genetic targets, and you have to evaluate those. But we have the tools to do that. The worst thing about an experiment is when you've got a sample of one. Dolly, our famous Rosalind Dolly, was a sample of one. From an experimental point of view, that's very little you can take from that, except that you can do it, all right? And that's what Dolly showed. So I like pushing technologies. Personally, I like pushing technologies to see what they can deliver. We can do it in livestock. We have engineered pigs for a virus called porcine reproductive respiratory syndrome virus, PRRS, and that is now in the commercial sector. The university has licensed that technology to a British company to see if they can develop a product from it. You can argue that we could go after other diseases in pigs or in other species, and we are doing that. And we have a large part of the effort at the Roslin Institute is looking at how we can identify. In the case of PRRS, it was a gene called CD163. It was exon 7 of CD163, which encoded domain 5 of the protein. All right. Fascinating to me, probably not many other people, but fascinating to me. We're looking for the next CD163 for another virus. It could be foot and mouth disease, it could be avian or swine flu, it could be classical swine flu, or diseases in aquaculture, livestock, farm animal diseases. And I am excited. I would love to find the genetic change I can do that gives foot and mouth resistance. I think it would be absolutely. We see that disease as an exotic. Many people around the world see it as their daily burden. It could transform milk production in India, for example. And that would be very exciting. And we are doing that. But in a way, it's not a frontier anymore because we've done the PRRS pigs. To me, the next frontier potentially the next frontier for using these tools is wildlife. And the way to get into that space is through gene drive. Not to say that gene drive is the solution, and it would certainly be absolutely staggered if it's a solution for many things, but it might be the solution for some. And pest control is the area that we're looking at. But it allows me to get engaged with different stakeholders, different opinions about whether or how we could envisage engineering wildlife. I go back to flu. So let's talk about flu for a minute, right? So obviously we catch flu. We pass it to each other, and that can be bad enough. But every now and again we pick up a strain from pigs or poultry, and it can be devastating in the population. If we understood enough biology, which we don't yet, we've got some candidates, but we don't have any sort of proof, and we're looking at those candidates. But if we understood the biology enough, we could engineer poultry, we could genetically engineer pigs, as the same way we've engineered them for PRRS to be resistant to flu virus. And that would stop that transmission to us, or at least dramatically reduce it. We could go one stage further and we could say, well, why don't we do it to swans as they fly over and fall into our ponds and spread the disease, or any geese that come flying in there, or anything else that comes flying in. So I think it's a really good question for us as a society to grapple with. Do we think it's acceptable to genetically engineer wildlife? We engineer nearly everything else apart from wildlife. We tell them where to go, we house them, and in many ways regulate wildlife. And if we are thinking that we could genetically engineer wildlife, what do we do with it? Would COVID-resistant pangolins have been a good thing to have 4 years ago? Right. These are really good questions. There are many answers and many ways to look at them, many lenses to look at them with, but I think we should have those types of discussions. So what is the sort of end point of gene drive? Well, I see two, when it comes to the squirrel project, I see sort of two end points. I do believe that when we understand and refine some of the designs, which is happening at the minute through all the great research in the insect community looking at gene drives for controlling mosquito populations, with that and with really extensive simulation studies which not only look at the impact on the grey squirrel, but also the impact on the sort of ecosystem that they reside in. I'm quite mathematical in my sort of logic behind things. The scenario where we can control numbers in a set context while allowing other areas to still have squirrels, I think is achievable and sustainable. The Squirrel Project, we have the opportunity of designing release strategies which will allow localized population control. This is not something you'd see for mosquitoes, not something you'd see for malaria, where you'd want to just eradicate the mosquito from the whole zone. So I think that allows us to bring in a refinement to the whole concept, which I think is exciting. And squirrels could demand that. Having said that, if you start to eradicate the grey squirrel from a population, and at least to a certain degree, and again we can model this, the red squirrel will still come back in over time. I think this allows us to have the debate and think about how you would put in place processes, both regulatory and physical on the ground processes, about how you would deploy these types of tools into wild populations. When it comes to the greys, the grey squirrels, they are, put it in context, they are slowly working their way up the country, have largely replaced any red that existed south of the border and are slowly doing that north of the border. There is the angle of looking after the reds in this, not only looking after or controlling the greys. So there are many different aspects that allow an endpoint which will inform on how we might, as a society, use these tools going forward. It's not just simply, this is a gene drive, put into that animal. And that's exciting because you don't know where that's going to take you. You don't know where that's going to end up, but you do know you will have the data and the understanding to allow you to have that discussion, allow you to have that dialogue about what might be a good. And I go back to this point where science goes on and it just happens. It happens everywhere, and there's a truth in that if you don't do it here, someone else will do it somewhere. When it comes to gene drive, we've got all the insect work going on. There are projects in Australia looking at controlling rodents, mice through gene drive. These things will happen, and I think it's good that we have activity here in the UK. There's another aspect, which is part of the reason why we're speaking, and that is that this is not simply a molecular genetic problem. It's a multidisciplinary project, and I really get fun out of that because you bring these different lenses, and you know how you look at the gene drive is subtly different to how I look at it, and that that adds a depth to whatever comes out of it, and that's good. That's good.